this is the best we can do at this point.
Okay,
Yeah.
I'm sorry.
No worries.
So we have our two different kinds of forces that we're thinking about. We have steric crowding interactions and non-steric sticking interactions or chemical interactions. So the steric interactions account for the rigid volume that the protein takes up in a crowded environment. And what this does is it favors reactions that increase the translational rotational freedom of a molecule by destabilizing extended unfolded states. And we can mimic this in vitro using inert macromolecules. So we'll often see people use PEG, and we like to use FICL to mimic these crowding interactions. And then we can also think about chemical interactions, or those surface interactions between molecules. This accounts for all of the interactions between the protein and the environment, aside from crowding. And this is highly dependent on the surface of the protein. So if we look at the difference between the folded and unfolded state of this protein here, we see that the chemical composition of the surface changes between these states, as does the charge. So there are a lot of different things that we can consider when we're thinking about what's giving rise to differences in chemical interactions in cells. The first thing that we can do is to change the ionic strength of our solution. So I think that this group is mostly theoretical, but maybe you don't appreciate for experimentalists, often a buffer is selected to stabilize the protein of interest or to be compatible with the experiment that you want to do. It's not actually selected to mimic what's going on in cells. So the first thing that we could do is change that ionic strength to match the cell. Then we could introduce some small molecules and metabolites, and we see that with our lysis buffers. So lysis buffers are used to lyse open cells, but what they're often designed to do is to stabilize protein-protein interactions for immunoprecipitation reactions. And so presumably that is also capturing some of these chemical interactions that are important for our proteins of interest. And then finally, we're also going to have interactions between macromolecules in cells, right? And so we can add some of this in by doing our experiments in dilute cell lysate. And so that's going to introduce some of those native proteins that would have been in the cell. And so I did a lot of this kind of work in Martin's lab. And one of the things that I ended on in his group, so this was published right as I was starting my independent position, is we looked at two different proteins. We looked at PGK, which is stabilized in cells, and this protein VLSE that is destabilized in cells. And we were able to find a mixture of our crowding reagent and our chemical interaction reagent that was able to produce the in-cell stabilities of both proteins. And so we suggested that perhaps this kind of mixture of 150 mg per mol FICL and 60% lysis buffer could be used broadly to predict in-cell behaviors. But we didn't actually test this, right? So as I was starting my lab, one of the things, or some of the questions that I had about this mimetic were, can this mimetic be used for nucleic acids? So nucleic acids are also in the same kind of environment. If we're capturing these steric and non-steric interactions, then maybe the same mimetic would work for nucleic acid. Does our mimetic work for peptides and small proteins? So smaller proteins don't encounter those crowding interactions in cells because they can fit in the gaps between the macromolecules. But we now appreciate that chemical interactions are really important. And so maybe this mimetic would capture those and be able to predict the stabilities of smaller proteins in cells. And then finally, I think what's really important for us to understand is, when does the mimetic fail? So does it always work? And it doesn't. So some of the newer stuff that we're working on right now are examples of when it fails. And I think that we have some interesting stories that I can tell you about. So the very first thing that we worked on in my lab is to look at the folding of an RNA thermometer. This was a great system for us to get started on because we needed to establish that we could build a fast relaxation imaging microscope in my lab. There'd already been some studies on this system, so we were comparing our data to some published data. We're able to reproduce that this RNA thermometer is slightly more stable in the nucleus of a cell than in the cytosol of the cell. And when we look at the distribution of stabilities of this RNA thermometer, we see that there's a lot of overlap, right? But in general, we see that things on average are slightly less stable in the cytoplasm than in the nucleus. And if we look at each individual cell that we analyzed, we see that in general, the trend across all of the cells is that this RNA is slightly less stable in the cytoplasm than in the nucleus. So there's a lot of heterogeneity, but that trend holds across all of the cells that we're looking at.
So
Yeah.
naive question from a computational person. The beta fluorophores are not affected by these conditions.
This RNA is labeled with dye, so it's ATO 488 and ATO 594. So you're asking, is it affected by the chemical interaction or like by our mimetic?
Or the salt, whatever you change the condition in your previous example.
Yeah, in our previous example. So we are able, so we don't see a big effect. If we change the label, so Martin has done some work where the fluorescent protein has been swapped out for dye, and that doesn't tend to affect the behavior of the protein. I have not tested the same protein with both fluorescent proteins and dye labels on it in my lab, but we are working on it to see if the mimetic will work. What I can tell you is that the same mixture will reproduce a trend on labeled and unlabeled samples. So that tells us that the behavior is not coming from the labeling itself, that we can reproduce these trends on unlabeled systems. So we're able to reproduce the in-cell stability of our RNA thermometer using that original mixture that we made, but we also wanted to be able to do measurements on the fluorimeter or by absorbance, and the lysis buffer was not optically transparent. So we tested another lysis buffer called mammalian protein extraction reagent. This is a lysine buffer, and this allowed us to do experiments on unlabeled and labeled systems. And we see the same mixture produces this same trend in both the unlabeled and labeled systems. So this gives us some confidence that what we're seeing is true for our system. So here we see 20% of mPER with 150 mg per mil of PEG reproduces our in-cell observations. We use PEG because there had been some previous work done with PEG in this RNA thermometer. And the reason that this percentage for the lysis buffer goes down to 20% is because of the strength of the detergent. So often the detergent is really important in these lysis buffers in determining how many of the interactions are broken. And so mPER has a more aggressive detergent in it than the lysis buffer, which is MP40, whereas the detergent in mPER is proprietary. So we know it's a zwitterionic system. So yes, we could use this mimetic for nucleic acids, and we see that we're now able to develop a version of this that's optically transparent. We then wanted to answer whether our mimetic would work for peptides and small proteins. And so we returned to a small protein that's been studied a lot. So one of Martin's past graduate students worked on lambda repressor. Actually, there were some of them working on it when I was in the lab. So we thought that this would be an interesting system for us to test. We could FRET label it. So we added those giant fluorescent protein labels to our lambda repressor, and we're able to see that labeling didn't affect the stability of our protein. So we're able to see that there's very little difference between unlabeled and labeled lambda repressor in vitro. We then moved our system in cells, and what we see when we move in cells is that lambda repressor is stabilized, and we also see that it's slightly compacted, or we see evidence that there's compaction. So our hypothesis for this kind of a system would be that the cellular environment is stabilizing lambda repressor using chemical interactions, because we didn't think that something this small, that's only about 80 amino acids, is going to be affected by steric crowding. So we made this observation, and then we return in vitro to try and explain our in-cell observation. And the first thing that we did is we looked at different amounts of our crowding agent and our chemical interaction reagent to see how that affects the end-to-end distance of our protein. And what we see is that as we add in our crowding agent, we're seeing compaction of our protein. And so this is evidence that either our protein is sensitive to crowding, or that we're pushing those giant fluorescent protein labels together, because those are 20 kilodaltons, and they're quite large compared to our protein that's really sitting as a spacer in between those fluorescent proteins. And then we see that the chemical interactions cause an expansion of our lambda repressor protein. So then we looked at the stability, And what we see when we measure the stability in all of these different crowding conditions is that there's no change in the stability of our lambda repressor as we titrate in increasing phycol concentrations. So this suggested to us that even though we're pushing the fluorescent proteins together, it's not affecting the stability of the lambda repressor. And so that this is just an artifact. Those lower D/A values are an artifact of being able to push the floppy linkers together that were on our fluorescent proteins. But we see that in MPER, we're stabilizing our protein. So we're getting this kind of a trend. And that same in-cell stability can be mimicked by 20% MPER alone. So the same percentage of MPER that we were using to replicate our in-cell RNA experiment was able to replicate what we see in cells for lambda repressor as well, with the additional piece that you can use MPER alone for the small protein. So the phycol is not doing much to the stability. So this tells us that chemical interactions alone can predict the in-cell stabilities for our peptides and small molecules. And now we get to this question of when does the mimetic fail? And so the previous two stories I told you have been published, and everything from here on out in my presentation is new data that's either under review or that is in preparation. So the first protein that I'm going to tell you about is Barnase. This is a really classic protein folding model. And the Barnase that we're using has a mutation to make it catalytically inactive. So this is a ribonuclease. If you express it in cells and it's catalytically active, it will kill your cells just by chopping up the RNA. And then we also needed to destabilize our Barnase so that it would fold at a physiological temperature. So we made a second mutation here. And what we see when we compare our unlabeled to our labeled Barnase is that adding those fluorescent protein labels onto this doesn't do a lot to its stability. So this gives us some confidence that we'll be able to do this in cells. We then measured the stability of our Barnase in the nucleus and in the cytoplasm of cells. And what we see in the cytoplasm of cells is that the protein is slightly stabilized, but that we're seeing aggregation concomitant with unfolding of our protein. So we're seeing this downward trend, which is consistent with aggregation upon unfolding. But when we look in the nucleus of the cell, we see that we're destabilized. So we're able to see this unfolding of our Barnase, and then we see aggregation occurring at a similar onset temperature to what we observed in the cytoplasm of the cell. Because we observe these opposite trends in the cytoplasm and in the nucleus, this suggested to me that our simple mimetic is not going to be able to reproduce this. We're not going to have some kind of condition with a crowding reagent and a chemical interaction reagent that's going to be able to mimic both of these. So we went ahead and we tested those, and indeed, just like phycol alone is stabilizing, changing the ionic strength is stabilizing, so just raising it from what people traditionally use. This chemical interaction mimetic has no significant change to the stability. It's when we start to look in lysates or in salmon sperm DNA, where we have non-specific interactions, that we start to capture these behaviors. So the fact that we're able to get a differential effect, that we see a slight stabilization in lysates made from the cytosol and a destabilization in lysates made from the nucleus that are reproducing this trend, that is suggesting that weak specific interactions with proteins in each of these compartments is actually modulating Barnase's stability. And now, Barnase is not a eukaryotic protein. It comes from bacteria, and so we wanted to be able to do our experiments also in this environment. So we made some measurements in E. coli, and we noticed that when we look at the folding of Barnase, that same one in E. coli, we see that it's destabilized by about 15 degrees. So this is similar to what we observed for our nuclear measurement. But what we also observe is that we never see any aggregation at higher temperatures. And we went ahead, since this was destabilized so much, we went back to the pseudo wild type, so just the catalytically inactive version, and studied this in cells. And what we see is it's destabilized by about 10 degrees in the E. coli. And again, we're able to get up to about 55 degrees, and we don't see any aggregation across these temperatures. So this is suggesting that proteins have evolved weak interactions to stabilize functional unfolded states in their native environment. And for RNase, unfolding is functional because this is a secreted toxin. So RNase needs to be unfolded and exported out of the cell where it'll fold and act as a toxin. If it was folding in the cell, it would, and catalytically active, then it would kill the cell. And so this brings me to kind of putting this in context, right? Like what's going on here? What can we learn about RNase? I was initially interested in just understanding this kind of from our chemical mimetic perspective and understanding the folding of small proteins, but I think we learned a little bit about the folding of RNase as well. So RNase comes from this organism where a inhibitor is also expressed, which is bar star, and it tightly bounds, or it tightly binds. So it has a nanomolar affinity. Because of this, it's thought to be weakly secreted, and then once it gets out of the cell, it can get into other cells where it will act as a toxin and be a ribonuclease, breaking up the RNA and killing the other cells. So this is our RNase and bar star. But when RNase was originally discovered and groups were studying it, one observation that was made was if you look at the catalytically inactive version of RNase and you look at the complex of wild type RNase with its inhibitor bar star, and you look at the amount of protein produced and the amount secreted, that there was no change in the secretion of RNase in the presence of bar star. And so this suggested to me that because the same amount of RNase is secreted with and without bar star, that this bar star is not related to the secretion, that there's another pathway or another mechanism involved in this. And now bar star may be a way to protect the cell if any RNase gets back into it, but it's not related to the initial protection, because if RNase was expressed and then you found it in the cell, it binds bar star, it's never getting out because it's not going to release that for export. So what we know, the other things that we know about RNase is it's often used as in complex with a chaperone. So it's been seen SecB, a secretory protein, and then also GRO-EL, which is a common chaperone. And these have been in vitro shown to act as unfoldases for RNase. So they promote unfolded states of RNase. And so our data really fits with this model. So what we think is happening is that there are chaperones in cells that are promoting unfolding of RNase, and this would allow our secretory proteins to transport RNase out of the cell where it can function. And if we go back to some of that original GRO-EL data, it was destabilizing RNase by about 10 degrees, which matches what we see for RNase in E. coli, which have GRO-EL. And so I think that our data on RNase are really exciting, and this is motivating us to look more at chaperone interactions with RNase in the future. So what we learned here is that weak specific interactions can modulate RNase's stability. And we've also found cases when we look at nucleic acids where our mimetic doesn't work. So we have been interested in the folding of I motif and G quadruplex DNA. So these are found in G and C rich tracts of DNA. And there's been even a lot of speculation about whether or not these structures exist in cells in native environments. So we see that we get these really interesting planar interactions between guanines and then a hemiprotonated cytosine interaction here that is going to stabilize these non-canonical structures. So can our mimetics predict the in-cell stabilities of these kind of structures? Well, we started by creating a labeling strategy. Here we use TAMRA and BAM, so small dyes to label our DNA. And we saw for G quadruplex that labeling had pretty much no effect on the structure. So this is circular dichromosome data that's showing there's no structural change. And it slightly destabilized the DNA. So what we see here is a melting of that. And then for our I motif DNA, we see that labeling doesn't have much of an effect on the structure of our DNA. But what we observed in phosphate buffer, which is true in most cases, is that our I motif doesn't fold at all. So for the I motif, often in order to get that hemiprotonated cytosine interaction, you have to lower the pH to promote folding. or it's been shown that crowding can induce folding of the eye motif. And so here we added 200 mg per ml PEG, and we're able to promote folding of that eye motif structure. And we see we get a similar stability between folded and unfolded eye motif here once we add PEG into solution. So we then wanted to make sure that we could monitor the folding of this DNA using FReT. And so what we see is that we can do that. So the stability of the G quadruplex is around 60 degrees in vitro, and our stability of our eye motif is around 30 degrees in the presence of PEG. So this would be our prediction for what we would see in cells, and we then do these same kinds of temperature-dependent measurements in cells. And what we see is that in cells, the G quadruplex is destabilized and the eye motif is stabilized in cells, and stabilized by quite a bit. So here we are microinjecting our labeled DNA into the cell. It will go to the nucleus on its own, and then we're just monitoring the kinetics and thermodynamics in the nucleus of the cell. So we wanted to use our mimetic to understand what's going on in these compartments. So why is G quadruplex stabilized? Why is eye motif destabilized? And are the same interactions giving rise to this? So we used our PEG, our crowding mimetic, and both of these structures are stabilized. And this is what we expect, right? Steric crowding stabilizes. We looked at our lysis buffer, and we see that both of these are destabilizing to both of our systems. So here red is G quadruplex and blue is eye motif. We then looked at some other conditions where we're seeing variable effects. So if we do measurements in DPBS, which is high ionic strength in sodium, we see that we actually do reproduce a differential trend. So however, it's pretty significant on the G quadruplex. It's highly destabilized, and it's only slightly stabilizing to our eye motif. So salt is really important in mediating this interaction. We then see that so in the nucleus, there's a lot of DNA. So we use salmon sperm DNA to see what happens with interactions with DNA, that this produces this kind of differential stability trend. So DNA is likely contributing to this differential trend in the stability in the nucleus. We see that protein, so BSA, if you add that non-specifically in solution, it's destabilizing to both. So just as a random protein in solution isn't going to produce any kind of differential trend. And we tested that because we almost completely recapitulate our in-cell observation here using cell lysates from the nucleus. So a nuclear cell lysate with those proteins extracted from the nucleus is able to produce this trend. So it suggests that interactions with nuclear proteins are really important to regulating the thermodynamics of the system. The mixture here is a mixture of our salt conditioned salmon sperm DNA, as well as our BSA, in an attempt to reproduce this, and it still doesn't get close to our lysate. So here, this nuclear lysate is really important. But I told you one of the reasons I really like fast relaxation imaging is that we can get kinetic information about folding processes, and I haven't shown you any kinetics yet. And so one of the things that I'm excited about in this project is that we've been able to measure kinetics in buffer and then in cells for each of these systems. And what we see is that for our G quadruplex, the folding is quite fast in buffer, and it's slowed down quite a bit in cells. So notice that the timescales are changed here when we move from in vitro to in cell. And we see the opposite trend when we look at the eye motif. So its folding is quite slow in buffer, but in cells, things are sped quite a bit. And we tested all of those same conditions that I showed you on the previous slide to see if, a lysate could reproduce the kinetics that we're observing in cells. And unfortunately, none of these conditions were able to replicate the in-cell kinetics. So you can see that there's an order of magnitude difference in the kinetics in the cell compared to all of these other conditions for our G quadruplex. And the same for the eye motif. It's much faster in the cell compared to in all of these different buffer conditions. So one of the things that we noted about our G quadruplexes and eye motifs is that they're compartmentalized. So we see them base separating in cells. And so perhaps this is important to modulating the dynamics in the cell. And we're able to induce condensation of our eye motif and G quadruplex in a few different ways. One of them is to add a positively charged protein like lysozyme. And here we saw aggregates formed. We got nicer condensates forming when we add polylysine. but I don't have a picture of that to show you today. I do have some measurements in poly-L-lysine, and what we see when we induce condensation is that our eye motif is stabilized, so its stability here is a little over 30 degrees, so not as much as what we observed in cells, but we're able to almost perfectly recapitulate the kinetics that we observed in cells. And so this is suggesting to us that not only are interactions with these positively charged proteins important for regulating thermodynamics, but this organization by condensation is also affecting the kinetics of our system. And so this is kind of where we are right now. This data was collected in the last week or so, but I think this is exciting and something that I'm hoping we're going to be able to see the opposite trend for our G quadruplex and be able to really explain what's going on with the kinetics in the system in addition to the thermodynamics. And so we see that our mimetic fails when weak specific interactions with these macromolecules are important for regulating the stability. And we also saw that compartmentalization can be important for modulating stability and kinetics as well. So we do have this mimetic, it's simple and commercial, and it can be quantitative in making predictions for in-cell trends, but if there are specific interactions, then a cell life state is going to be important. So I wanted to use the rest of my time to talk about some of this unrelated stuff that we're working on. So fluorescence imaging and fluorescent proteins have revolutionized our understanding of the cell, right? Using these dyes and fluorescent proteins, we can track kinetics and thermodynamics in real time. But fluorescence microscopy isn't perfect, right? Fluorescent proteins can crowd, self-associate, and interact with a protein of interest. Our dyes can cause mislocalization. They're difficult to incorporate, and they can be difficult to introduce into the cell. And if you're interested in metabolism, a labeled metabolite isn't going to fit, right? You have a lock and a key, and so you're not going to get proper metabolism. So we want label-free, or as close as possible to label-free, structural and functional information inside cells. So is vibrational spectroscopy an answer for this, right? My background is in IR spectroscopy and IR imaging, and every molecule has a unique IR signature associated with it. So we have glycogen, lipid, DNA, RNA, protein, and all of this is going to come together to build the IR spectrum of a cell. So it's going to be the combination of all of these components. An issue with IR imaging is the diffraction limit. So when you're using 500 nanometer light, you're diffraction limited at around 250 nanometers. You can see organelle inside cells. When we expand this wavelength of light out to the mid-IR, we're now diffraction limited at around 10 microns, and it's going to vary depending on what frequency you're using, right? Because you're using a bunch of different wavelengths of light. Now you're only going to get 1 or 2 pixels per cell, so you're not going to have that spatial information that you had before. So we are using optical photothermal infrared imaging to break the diffraction limit in the IR, to provide the spatial resolution of our fluorescence microscope with the information from the IR. So this is a pump probe technique. We pump our sample with a tunable IR laser, and we probe with a visible laser. As that IR laser is pulsed on and off, the sample is going to thermally expand when it absorbs that light. That changes the index of refraction at the surface of the sample. We can detect changes in the scattering of our visible light and use that to back calculate how much of the IR light was absorbed. So now we can get full IR information, but because we're using the visible light to image, we're now diffraction limited in the visible again. So this is a trick to get us this high spatial resolution IR information. And one of the first things that we thought about was, can we correlate our IR data with fluorescence data? So we have fluorescence histology stains that can be used to identify different compartments in the cell. And this should have information about all of the different proteins and molecules that are contributing. So each organelle will have a unique spectral signature. And we were able to collect these kind of hyperspectral images and train a computer to identify all of these different compartments. So we have nucleus, nucleolus, Golgi, ER, mitochondria, nuclear speckles, and we can use random forest or UNet neural network models that have been trained on this data to then make predictions and accurately identify these compartments. So now you could do histology without ever needing to stain your cell. But you're not just getting that, you're getting additional information out. So if we look at stress granules, for example, and we're interested in the protein composition, what we see when we look at the cytoplasm in the nucleus versus the granule, this is the amide 1 and 2, so this is telling us about protein structure. What we see is there's a little peak here in orange that's not in the other data. This is beta sheet content, so it's telling us our stress granule is rich in beta proteins. So we're getting compositional information. We're not just getting yes, no, there's a stress granule in there. We're able to say something about the composition of that organelle. But I want to be able to track specific proteins of interest, right? I don't want to be looking at just the bulk signal. And so we need probes that we can introduce to track these biological processes. And luckily, IR spectroscopists have provided us with small genetically encodable IR probes that we can use. So some of the ones that were developed in my grad work is we use azidohomoalanine, which is a methionine analog developed by Carolyn Bertozzi to track protein folding, so that azide is sensitive to its environment and it will change its position in the folded and unfolded state. So we can use that to track folding of a protein. I used isotope labeling to label a single methionine in a protein and track folding changes. So using this single isotope introduced into an individual methionine in each helix, we could say something about which helix was forming first in this protein. Wei-Min has nicely showed that these same probes can be used in whole cells. So here we're seeing some work that he's done with azide labeling and azidohomoalanine, as well as with isotope labeling. But a limit of this work was the spatial resolution of the instrument. So can we use these kind of probes with our O-PTIR imaging approach? And we've learned that yes, we can, but not all of these probes are going to be compatible. And this is because we need probes that are not only going to be IR stable, they also have to be photostable. And some of the IR probes are photoreactive. To show you that, I have one of our favorite proteins to test, superfolder GFP. This is nice for testing incorporation of unnatural amino acids because if the unnatural amino acid is properly introduced, you'll see fluorescence. So this is a fluorescence image, and we can collect fluorescence images on our O-PTIR microscope because we have visible light. We then can take a full IR spectra of E. coli, and here we see in a normal E. coli expressing SFGFP, we have protein signals, so amide 1 and amide 2, and nothing in the cell silent window. We can then feed our cells this azidohomoalanine and a methionine and auxotroph cell line. So these cells don't natively have methionine. They can't make it themselves. You have to give it something to replace that. So if we feed them azidohomoalanine at induction, we see expression of our protein. So this will incorporate azidohomoalanine at all 5 of these methionine positions. We know that it's been incorporated because we see fluorescence, and then we image or we collect a IR spectra, and we can see that azide lighting up there. So we know we've successfully incorporated this. But this azidohomoalanine is incorporated non-specifically into all methionines in our cell, right? We want to be able to look at a specific protein at a specific position. And so we can use amber codon suppression to introduce an unnatural amino acid here at this valine position. And this is azidomethylphenylalanine. We know that we successfully express it because we see fluorescent E. coli, and we can detect that single amino acid incorporation in our E. coli. We can see that here. So we have this in the cell silent region. But if we use a photoreactive probe, like azidophenylalanine, which is used for photocross-linking mass spectrometry, we know we can incorporate it because we see fluorescent protein. But when we try to use O-PTIR to image this, we then react it and we're not detecting anything in the cell silent region. So this was an important lesson for us in using this kind of instrumentation. We also know that O-PTIR can track metabolism in cell line specific ways. So if we feed our cells C13 glucose, it's going to metabolize that glucose in a way that's dependent on the cell line of interest. So if we feed C13 labeled glucose to E. coli, it's going to use it differently than if we feed it to fat cells. So we fed C13 glucose to these 2 different cell lines, and anything that shifts over the time, so over 72 hours here, is associated with metabolism of glucose. In E. coli, we're seeing shifts of the protein bands. In our fat cells, we see shifts of the ester carbonyl of the triglyceride, so it's being used to produce fat. And I haven't shown you any images from our microscope yet, so here are some images. This is a fluorescent image of a single E. coli, and then we have our fat cell. And when we create a ratio image collected on our microscope, so ratioing the isotopically labeled to the C12, we can map where this has been incorporated in the cell. So the protein in E. coli is mostly in the center with less towards the extreme here. And then we nicely detect that C13 is incorporated into triglycerides that are in our lipid droplets in the cells. And the other thing that's interesting here is that it's pretty homogeneous. So we're not seeing a lot of heterogeneity across these different droplets. So we have been interested in this lipogenesis process because non-alcoholic fatty liver disease is a global health crisis affecting about 25% of the world's population. So the dysregulation of lipogenesis is a significant factor in this disease, and you can't study this in a test tube. You need to consider all of the enzymes that are a part of this. So we have glucose going through glycolysis, the TCA cycle, and then de novo lipogenesis, where these lipids are ending up in triglycerides. And your diet is going to play a really important role here. So you have free fatty acids then that can be esterified, and your saturated free fatty acids are going to promote disease and death of your cell, and unsaturated free fatty acids are protective to the cell. So it's unlikely that a single enzyme here is going to mediate disease, and we need to be able to observe this, in context. So the first system that we studied was the metabolism of oleic acid and its effect on the cell. So oleic acid is protective, and there's two opposed mechanisms. One is that it can directly inhibit acetyl-CoA carboxylase here. A second is that it decreases expression of sterile regulatory element binding protein 1C, which is a transcription factor for all of these proteins involved in de novo lipogenesis. If you knock out that protein in a liver cell, it will die, but adipocytes won't. And so our hypothesis was that if we saw a differential effect in the two cell lines, this would support the second mechanism, where it was SREBP1C that was playing a role. So what we do is we feed our cells C13 labeled glucose and deuterated oleic acid concomitantly and observe how those are metabolized over time. We grow our cells on calcium fluoride cover slips and create an imaging chamber using a 5-micron spacer, and we image our cells in transmission mode. And then we take images at specific frequencies of interest that we observe in the full spectra. So the protein, that CD stretch, and then our C13 and C12 labeled lipid. When we do this, we can nicely detect in both our liver and in our fat cells that CD stretch, as well as the C13 glucose incorporation, this little shoulder over here. And we first measured the basal rates of both of these cells, and we see that both fat and liver cells are metabolizing the C13 glucose at similar rates. So this is just the rate of incorporation of C13 over time. And then we can look at what's going on with the deuterated oleic acid. So these are ratio images of image collected at the CD stretch to background lipid. And what we see here is color indicates how much of this has been incorporated. So at 24, 48, and 72 hours, the color is very similar. This is telling us that our free fatty acids are metabolized really quickly and then stored. So we went and looked at earlier time points, and we can see that we're quickly metabolizing our oleic acid. We can detect it within 2 hours, and then the amount in cells is stabilizing around 24 hours. We can do this at the same time that we feed cells C13 labeled glucose, and we see this is metabolized more slowly over 72 hours. Just to highlight, we can only see these probes in a lipid droplet. So this is a full spectra and a lipid droplet versus in the background of the cell, and we're only seeing these stretches I'm assigning in those lipid droplets. This is quantifying what you see over here. So we're not seeing a big change in the amount of deuterated oleic acid in time, but we're seeing that C13 glucose being metabolized slowly. We did the same thing in a fat cell, and what we see is that in the fat cell, overall, there is less oleic acid in our lipid droplets here because of the increased background lipid to begin with in our cells. But things are, again, metabolized very slowly. And so if we compare our liver cells to our fat cells, what we saw is that there's a statistically significant suppression of de novo lipogenesis in our liver cells, but not in our fat cells. And we were concerned that this had to do with the change in morphology of the cell. So you can see when you feed oleic acid, you get these really big lipid droplets that you don't see in the control cells. So we controlled for that. And what we see is that the rate of change is different in our C13, our oleic acid fed versus in control. And this gave us confidence that it really is a suppression, that it's not an artifact of the increased lipid. So if we go back to this supports a model where SREBP1C is regulating de novo lipogenesis. Now, this might not be as exciting as our saturated free fatty acids. So why are these promoting disease and death, right? We can do the same kind of thing using deuterated palmitic acid. So why is palmitic acid toxic to cells? Is it upregulation of de novo lipogenesis? Is it increased circulating free fatty acids? Well, if we reproduce the same study I just showed you with palmitic acid, we see that like oleic acid, palmitic acid is suppressing de novo lipogenesis. So this suggested to us that it must be free palmitic acid and palmitic acid intermediates that are leading to this lipotoxicity. And so we can do the same thing. Here we don't even need to worry about de novo lipogenesis now. Let's just think about what's going on with palmitic acid. And we can detect this upon feeding. But what we notice is that there's a really cool shift here in the data of the ester carbonyl upon palmitic acid feeding. So if we expand this out, there's a shift from 1744 to 1734 upon palmitic acid feeding. This is deuterated palmitic acid, but this is a CO stretch, so it should have nothing to do with that CD stretch. So I said, just feed the cells palmitic acid and see if we see a shift there as well. And indeed we do. So we see the shift in the position of the ester carbonyl of our triglycerides when we feed palmitic acid. And so I said, okay, I think that we're seeing an overload of unprocessed fatty acids, even at the 24-hour time point that they're building up in the cell. So we went ahead and we looked at different time points, and we can see that this reaches a maximum, the shoulder, at 27 hours, and then it disappears at 70 hours. So this, to me, really suggests that this is a metabolic effect that we're seeing over this time period. You can take the second derivative to help enhance these bands if you want to assign them. So what's going on with palmitic acid metabolism is that you have palmitic acid and you have a glycerol, and you need to attach 3 copies of that palmitic acid to the glycerol to build your final triglyceride. And all of this chemistry is happening in the ER of the cell. And there have been some nice mass spectrometry studies that show, so this is a control cell versus palmitic acid fed. The color is indicating saturation, so you get a buildup. The size here is the amount of these intermediates upon palmitic acid feeding, and they're saturated. So this suggests that what we're seeing is precursors of our triglyceride upon this palmitic acid feeding. And I said, okay, well, every molecule has a unique spectral signature, right? That's what makes IR spectroscopy cool. So let's go ahead and look at the IR spectra of these different intermediates. Can we assign it? And what we see here is that this 1734 band lines up really nicely with the diacylglycerol with a DAG. It's definitely not palmitic acid, which is down here. These others are kind of close together, but it seems to give some confidence to what we're seeing is either palmitin, which is a MAG, a mono acylglycerol, or this DAG here. So a single copy of palmitic acid or 2. We also had seen some literature that had done some TLC plates. This like threw me back to organic chemistry. I had not done this since then, where they were able to show that there is DAG buildup upon palmitic acid feeding. And I had a grad student who willingly replicated this work and was able to show that we do indeed see a buildup of these DAGs upon palmitic acid feeding. This gives us more confidence in our assignment. But the power of this approach is that we also get spatial information, right? And I'm telling you, this chemistry is happening in the ER. So what we see when we do a line scan through a lipid droplet is that there's also a spatial component to the location of the shoulder. So color here goes from orange to kind of blue. So we see the shoulder, as you go from orange to blue, is shifting over. So this intermediate looks to be at the interior of the cell. But we also have the ability to do fluorescence experiments. So we stained for lipid ER and nucleus, and then we zoomed in on one of these regions of the cell where we have ER and lipid and created a ratio image of that triglyceride ester carbonyl to the shoulder that we're seeing. And what is highlighted really nicely here, all of these bright spots are actually in the ER that is adjacent to the lipid droplet. So we're able to see palmitic acid intermediates building up in the ER of the cell. And this is completely label free. So we don't even need the CD stretch here to be able to see this. And if you're thinking about Raman imaging, an advantage of this over Raman is not as sensitive to the carbonyl stretch as the IR is. So we're actually able to make this assignment that we wouldn't have been able to make just using Raman, and those CD stretches look the same by both in all of these intermediates because they're the same length, right? So we really need to be able to see the head group and able to order to make this assignment. Okay, so I've told you a lot about what we've been up to in my lab, and hopefully you think that some of this is exciting. I'm definitely excited about it. Everything's really coming together. I showed you that some of these simple mimetics can be used to reproduce in cell stabilities, that lysates are important for specific interactions, and we have some evidence that these interactions can really be functional for the native environment. I showed you that this O-PTIR can be used to track competing metabolic pathways, and that we're able to provide support for mechanisms that have been posed in the literature. Our oleic acid data suppresses SREBP1C and not acetyl-CoA carboxylase. And then we see that free palmitic acid is esterified slower than oleic acid. We see a buildup of these intermediates, and that this may be part of the lipotoxicity of these. But like oleic acid, palmitic acid suppresses de novo lipogenesis. And with that, I would like to thank the people that contributed to this work. This is my lab, so as Martin said, it's gotten really big. I have 11 graduate students and 1 postdoc right now, and I highlighted the people who did all of the work on the side. And this work is funded by the NIH and NSF, as well as private foundations. The Beckman Foundation, Evolution of FAR, and the Coastal Community Foundation have funded this work. And with that, I'd be happy to answer any questions.
Seeing Doug's name there reminds me of something.
Yeah, this is my summer project.
I'll patiently wait. Questions?
So 2 questions about the last. Sorry, I have to.
Yeah, it's okay.
5 minutes. So what are the limitations or in terms of imaging molecules? Are there any molecules that we cannot see?
Yes. So IR is concentration limited. So you're looking at needing to have, I would say millimolar. I think if we really wanted to get, maybe we could detect 100 micromolar. There are some groups using fluorescence to enhance the signal, but just using the pure IR signature of a molecule, you're limited, similar to like an NMR experiment in traditional IR. And the other limitation here right now on our microscope is speed. So we're using a stage to scan, and if we wanted to collect a full hyperspectral image of a cell, it would take like 12 to 18 hours. So for just a single frequency image, we can collect that in about a minute. But when we're thinking about I'm really interested in protein dynamics, that's quite a bit faster. This is really just providing an equilibrium measurement in those cases.
The molecules move inside the cells, and we try to localize them to find something that doesn't move. Right?
Yeah, so luckily we need to find things that aren't moving and that are condensed. And a lot of the condensates aren't moving a lot. And so that has provided us with this ability to do this kind of imaging in the lipids. Yeah.
Kind of bouncing off of Ahmad's idea there. Have you ever tried so when you do these ratio images, have you ever tried collecting the exact, do you guys try collecting the exact same frame multiple times to see, do the lipids redistribute in the time frame of you collecting them?
Yes, we have, and they don't. So we've done experiments where we collect a single scan and then collect another one back to back, take the difference between them, and the difference is minimal. Yeah, so I don't believe that to be true. We know molecules are dynamic. Yeah. It's going to depend on the organelle that you're interested in, right? Yeah. And for example, for E. coli, everything is kind of in that one spot, so that makes it nice for us too. Yeah.
Did I get the message right that when you are creating the say mimetic for the cellular content, you always need some cell lysate.
You don't. So, okay, so I think if you don't know anything about your system, that cell lysate will give you a accurate prediction, because sometimes interactions with macromolecules are important. But what we found is that often just the lysis buffer itself is enough to replicate a lot of the chemical interactions. So only in some cases is the lysate important. And the reason that I don't want to use lysate is because it's difficult to prepare. Like when I do those experiments, I fill the whole incubator with cells to make the lysate. And production of lysate, it's difficult to get batch to batch, there's a lot of heterogeneity batch to batch in it, right? So it'd be better if we had a simple mimetic, and that simple mimetic does do a really good job in general. It's just these cases where there appear to be some specific interactions that are important in the cell where you need that. Yeah.
Thank you for the great talk. Going back to some of your azeto labeled photothermal data, I noticed in the molecules that had the azeto label, the amide 1 amide 2 bands looked a little bit odd in the spectrum.
Yeah.
Is that just due to the label?
So yeah, in this data, these are dried cells and they were dried to a different extent. So I know exactly what you're talking about.
Yeah.
So it's just these were actually dried, and water overlaps with the amide 1 and 2 in cells. And so if you don't fully dry it, you're going to get an overlap. And so that's what you're seeing a little bit of here. Yeah.
So it's not.
Yeah, it's not inherently related to that. What we do see that I think is important is that like sodium azide is often used to stress cells, right? Like azide is toxic. We do see a lipid in our especially our azidohomoalanine fed cells. That's real, and that's indicative of cell death. So it is it's not just a silent observer methionine, like it's doing some other things that are toxic to the cell that eat it. Yeah.
Yeah, like in vitro measurements, you can just store as much vitro as you want. So
Yeah.
you get a lot of that.
Yeah. So what we found for azidohomoalanine is that it basically completely shuts off the metabolism. If we try and like continue to grow the cells for days, they definitely don't like having a lot of that around in the cell. Great question. Yeah.
So back to the coordination methods, how is that versus because I remember we used to cherry that you can just like, it's as simple as that you just have like the fluorescent protein cell and change temperature. The fluorescence will depend on
Oh, so you're asking, is there a temperature dependence?
Yeah. I guess my question would be using FReI would be maybe like more accurate or more.
Okay.
The slope would be like more dependent on temperature. Or
Uh,
would you even do that, I guess?
Okay, so anytime you pick a fluorophore, whether it be a fluorescent protein or a dye, it's important to characterize the temperature dependence of that if you want to do a temperature dependent measurement. Atto and Alexa dyes were actually designed to have pretty low photo bleaching and temperature dependence in them. So they're pretty photostable in general compared to some of the others. I showed you Tamara FAM as well. That has a strong temperature dependence to it. So the data need to be corrected for that when you're working with it. But you would do the same thing that you do. So mCherry and GFP also have some temperature dependence of the quantum yield. And so you do experiments with those probes alone to learn about their behavior and then use that. So what we see is that over like short temperature ranges, things are linear, and so we can approximate them in that way and then extract our kinetics from that.
I also noticed you were going to high can really go to 6 degrees Celsius.
mCherry is stable up to 94 degrees. Okay. So it's your organism. Yeah, the cell is the limiting factor. So it's just whether or not your cells are dying when you're going up to those temperatures. Yeah, GFP unfolds at around 75. So like, getting up in high in temperature, you may start to unfold that. And we've seen that before. But again, like all of that is just in the characterization.
Well, let's thank Casey again for a great lecture.